Up next, we have Adrine de Souza. Adrine is a PhD student at the University of Toronto in the Department of Molecular Genetics. She's supervised by Fritz Roth. She's leading a large-scale variant effect testing for VHL in AGXT genes using VEMP-seq and yeast complementation assays. She's providing evidence for variant interpretation of renal cell carcinoma, VHL disease, and primary hyperoglycuria type 1 patients. Adrine completed undergraduate studies in biotechnology in Brazil and participated in an exchange program in Boston. She's interested in scientific communication, implementation of evidence-based care, and teaching. So whenever you're ready, Adrine. You're muted.
Hello, my name is Adrine. I'm from Fritz Roth Lab. And although I have projects to evaluate variants of HXT and VHL, because Megan actually presented about VHL today, I'm only going to focus today on my large-scale testing of HXT related effects. And both of these genes actually affect renal disease. So the strategy that we are using to test variants in large scale is called multiplex assay of variant effect, or MAVE, and I'm going to introduce you to this concept today. So before we can start talking about the strategy to test variants, we have to talk about the crisis. And the main problem today that prevents us from having a real precision medicine is that we cannot interpret variants fast enough. So if a patient gets genetic testing and a variant is identified, we ask, is that variant pathogenic? Does it really cause disease? And you can actually appreciate by this plot here from variants in ClinVar how the number of variants of uncertain significance has skyrocketed in the last decades with more availability of sequencing. So instead of having a label of pathogenic or benign, actually most of the variants today have no evidence for their function. And this is a problem because without this type of evidence, patients cannot benefit from a definitive diagnosis, from cascade screening or other interventions. So there are actually different levels of evidence that could provide variant classification. And here I'm showing you the guidelines of the ACMG-AMP group. And I'm highlighting the important evidence for us here in this talk, that is the functional data that can provide strong benignity and pathogenicity evidence compared to predictive data or population data and co-segregation data. So to really test variants in a more parallel, a complete way, we have to go from the single variant testing that we'll call a reactive testing. So in this case, if that variant that has, if that patient that has that variant is in the clinic and that variant is identified, there may be the possibility of single variant fitness test, and maybe that could lead to a different variant annotation, but we call this a reactive testing, because if we wait for that variant to be detected to test it, you can imagine that we won't have enough time for, let's say, preventive measures, considering we have almost 1 million of VUSs. So you can see that this can take a long time, and patients might lose the window of something they can do to decrease symptoms or prolong life expectancy. So what we are suggesting here is a change of paradigm to really retire the reactive testing and have a proactive testing approach, where we actually test all possible variants in parallel. So we call this multiplexed. And we can get the experimental evidence for function of thousands of variants of a single gene. So we actually make these lookup tables that we have already detected functional variants even before they are detected in clinic. So then I'm going to introduce you to the gene that I'm working here that is called HXT. So the alanine dioxylate alanine transferase is the HXT gene and encodes for the ADT protein. So here I'm showing you the monomer of ADT that has an N-terminal domain, a large domain, small domain, and C-terminal domain. And I'm also highlighting here the cofactor binding sites. So the active form of vitamin B6 is PLP, and is actually a recommended treatment for patients that are affected with this lack of AGT. And once this protein dimerizes, it transforms glyoxylate and glycine in the liver peroxisomes. And I'm highlighting also two residues that are really important for this project. So the main problem here is once AGT is not active in the liver peroxisomes, we see a buildup of glyoxylates that in turn will become a buildup of oxalate. And this leads to kidney disease and kidney stones. And the name of this rare disease associated with AGT malfunction is primary hyperoxaluria type 1, and is autosomal recessive disease that affects 1 to 3 in a million per year. And by age 25, 90% of patients are actually symptomatic. And 90% of patients will have complete loss of their kidneys by age 50. And patients with this disease need kidney and liver transplant because they need to fix the problem in the liver to not have another kidney affected. And here I'm showing what is the main problem with AGT malfunction. It's actually mislocalization. So I'm showing you here in the left panel the dimerized form of AGT, and I'm highlighting in red some residues that are actually disorder in the minor allele. So AGT comes in two forms: the wild type form that doesn't have any different residues, and the minor allele mutation that is present in around 5 to 20% of people. And this minor allele has a leucine instead of a proline in the residue 11, and has a methionine instead of isoleucine in the residue 340. And what happens is actually the wild type protein can just folds, dimerize, and goes 100% where it's supposed to go. But the minor allele mutation actually makes a very weak mitochondria targeting sequence. So this protein ends up going to 5% to the mitochondria and also has some loss of function. And then this doesn't necessarily cause disease, but other mutations that are insist with the minor allele. So here is a PH1 example variant, GLI107RH. 90% of this protein, if in the minor allele background, goes to the wrong place. And then we start to see symptoms and rare disease. So here we are taking advantage of a yeast complementation assay to really test human variants. So our collaborators engineered this yeast that lack 4 different genes that are important for glycine metabolism. So this yeast cannot make glycine, and AGX1 is the yeast ortholog responsible for glycine. And then if we add this yeast strain into a plate that doesn't have glycine, here show as the empty vector, this yeast cannot make glycine, thus cannot grow, so they die. But if you add back either the yeast ortholog, AGX1, or the human AGXT, you see more growth because these yeast are being able to make glycine and survive in this media. So then, to make sure that we could go large scale, I needed to know that we have enough pathogenic and benign variants for validation of this assay. And here for AGXT, you can appreciate how we have pathogenic and benign variants across the entire protein. So then I went for the pipeline of MAVE to really test all the missense effects of AGXT by first doing mutagenesis of all possible missense variants. And then I make this library in the background of the wild type allele and in the background of the minor allele. And I transform this yeast and I allowed them to grow in media either without the cofactor or with cofactor treatment, so plus PLP. And the idea here is that just allowing this yeast to grow in media without glycine, only the variants that could make glycine would allow the yeast to grow. And then we see more frequency of those variants after the selection. So then I use the samples post-select compared to pre-select to sequence the variants, calculate the frequencies, and then by having a ratio between the frequency, I can calculate fitness scores. So my goal is actually to make 4 different maps for HXT in 2 different genetic backgrounds and 2 different context or treatment cofactor contexts. But going a little further on how we calculate this fitness scores, I'm showing here an example of yeast that either have a nonsense mutation that has a termination on that site, a wild type variant, so here the yellow that just grows normally, and a red variant that works better than wild type, better than expected. So you can see that after selection, this difference in frequency can actually be measured with a number based on the frequency of this variance. And based on this ratio, we make functional scores. And we translate this into a color scheme that will have a functional impactful variance as blue, yellow is wild type. We can even detect variants that work better than wild type in red, and white variants are actually neutral-like variants. So then we actually plot this based on all the possible missense mutations and each one of the residues, and we do this for the entire protein. And here is the very exciting results. So far, I have 3 maps for AGXT variant effect. And you can see that the title of my presentation actually has to change a little because now I also have the PLP cofactor treatment context for this protein. So then we have 3 maps. The first one is all the variants in the wild type background, and you can see that there are spots, they're very dark blue, so they're very intolerant to mutation. And for this map, I cover 98% of all possible variants without PLP, and then once I add PLP, I was able to get around 84% coverage. And you can appreciate how many variants that look like they're working better than wild type actually starts to appear when we have cofactor. And even the blue regions are a little bit less intense. So it means that cofactor could be helping this variants to work better. And then at the bottom part, we have the variants in the minor allele background without the presence of PLP. And I think it's really important to mention here that I detected a very high number of frame shifts around this region here that are also known to be a substrate binding site. And this last region of the minor allele actually shows a lot of variants that are working more than the wild type. So we want to make sure that this is a real finding. So I'm going to retest this map to make sure that this is actually maybe an adaptation or if it's real. But then going to more the data analysis of these maps, I'm here first showing you the separation between the synonymous variants, they are the ones that have the same amino acid, and the nonsense variants, they are the terminations. So they are in red. And you can see that in both major allele maps, with and without the presence of the cofactor, we see good separation between the synonymous and the nonsense. And actually, you can see the missense that I tested in gray here in the bottom part for all the 3 plots. And the separation of the minor allele is not very good. So that's why I'm retesting to make sure that I can separate better this nonsense variants that are looking like synonymous, but it might be just an adaptation of this variance and this yeast to the minor allele. So this is something that I wanted to study more. And then here's another way of showing the synonymous and nonsense separations just based on the plotting of the fitness scores. And I hope you can appreciate how all 3 maps can really separate what is functional and what is not functional. So this is really exciting as a benchmark for us. And it's also really interesting to see that the protein actually stopped having very important residues at the end, actually around 9 amino acids. Nonsense start to have synonymous like function. And again, the minor allele has some difference in their like last part, the tail of the protein, the C-terminals, that we would like to see if these nonsense scores are looking a little bit better actually behave that way. So now that I have these 3 different maps, and they are all very representative of all the possible missense variants of HXT, I wanted to ask questions about it, right? Because now we can ask many biochemistry questions and get answers using this data set. So first of all, I wanted to explore the important residues for function. So here I'm showing you in the wild type background map without the presence of PLP, some regions that look very impactful or very intolerant to mutations. So if, as an example, in this black region here, you mutate any of these amino acids, they look very dark blue. And actually this residue here is interacting with the active site that here is in purple that binds to the PLP cofactor. So we can identify these regions as very important for function. And the same thing happens to other regions of PLP binding sites that are very impactful, and to residues that are known to have some type of homodimerization importance or function. So then, could I look at the major and the minor allele scores and see if there's any difference between the two proteins? Yes. So here I'm showing you the major map on top, the minor map on the bottom, and again, highlighting the region that I found a lot of frame shifts. But then I did a moving window correlation analysis using these two scores. And the red one, that is the major scores, and the blue one, that is the minor scores, actually have very similarity throughout the protein, even though the minor scores look a little bit lower. That makes sense with the biology that indicates that the minor allele is at the edge of the function for the protein. But some residues that are really interesting to look at, as an example, the active site here that binds the cofactor, the scores are very similar between the two maps. And also there are some residues that are interesting for us, and we are doing many different analyses, including molecular dynamic analysis, to make sure that this that we are seeing is a true difference in the two proteins. So finally, can we look at how PLP affects the scores of the two maps? Now we can, and here I'm actually highlighting a disorder region that is known to be disordered in the AGXT major and minor allele, but as more disordered in the minor allele, so here in red. And this disorder region actually, when I compare the moving window analysis between the major in red without PLP and the major scores with PLP in blue, only this region that is disordered have very similar scores of the two proteins. And there is an indication here that if we add back PLP, so if we add cofactor as patients have vitamin B6 treatment, we actually get a little bit better scores. So then I wanted to talk a little bit of the validations we are doing with these maps and what type of analysis and computation predictors actually have any correlation to our scores. So as a background, computational predictors, they have a wide range of evidence strengths. So it changes related to the methods, but they could be also used to annotate or to reclassify variants. And so far, I'm actually showing here the most up-to-date, the top current computational predictor that was just released this month, alpha missense. And they compare if they trained on the CleanVar data sets, can they predict fitness scores or pathogenicity scores for variants? And alpha missense has showed a very good area under the curve, more than variety. So I just wanted to show you this because I first used variety, that is a predictor that was made in our lab, and ESM1B, that was the second most top predictor before alpha missense for my latest validation. So I'm really excited to also look at alpha missense now that this is available. So just talking a little bit of the predictor. So I talked to you that I looked at ASM1B, and here I'm doing a correlation analysis of the scores of ESM1B in red and the scores of the major map without PLP in blue. And you can see how the peaks and the valleys are actually very similar across. So maybe this is indicative that our scores also have a very good correlation to pathogenicity predictors. So then I wanted to show you some other types of validations, now getting to the phenotypic data and the annotation part of the project. So here is a genetic testing company called Invitae that they do have their own system to annotate variants. It's called a Sherlock, a point-based system. So they actually called benign variants based on their system as the green circles. The pathogenic variants that they call are red triangles, and we have the VUS. And if I plot all the fitness scores for each one of these variants, we actually see that there is a segregation between pathogenic variants having lower scores, benign variants having higher scores. And we can actually make a line there around 90% of precision for us that we can separate the variants based on function. So this could be used in the future to maybe reclassify variants. So here, another analysis for us is the precision recall curve of the maps that have the scores in black. The Verity that I talked that was like the second best predictor in red. And yes, I even did analysis with ESM1B that have the green lines. So here in the left panel, I have the precision recall curve for the wild type background. And in the right panel, I have the same thing, but for the minor allele, and I haven't used yet ESM1B for this. And the important thing is that we are using as the reference set here the same Invitae pathogenic and benign annotative variants. That is the largest cohorts for me for a validation of the experiment. And you can see that if we make a threshold around 90% precision, we get scores that have a good area under the curve. So that's really good for both maps. And we know that if we set a recall that is all true pathogenic variants, how many are observed as damaging, we get around 70% precision for the major map and around 80% of precision for the minor map. And then using this type of reference sets here, so now the benign variants are the green lines and the pathogenic variants are the red lines. And I can actually make a measurement of log likelihood of pathogenicity using the fitness scores. So using this, I can actually separate levels of evidence for each one of the variant types and how could that be related to disease. So in this example, we have a good separation for the major allele, and we actually notice how benign variants are looking like having a worse score in the minor allele background, which makes sense because the minor allele sensitize the protein to other variants. And then finally, I looked at the ClinVar database, and I was able to look at all the AGXT-VUS missense there are 50, and we produced some level of evidence for 37 of them. So here you see from a range of very strong, pathogenic, benign, strong, to supporting and moderate. And finally, I have the conflicting annotations on ClinVar, and I looked at the log likelihood of pathogenicity in the y-axis and the fitness scores in the x-axis, and we can see that there is a class of variants here, the yellow, that have the conflicting of VUS or either pathogenic variants that have a very high likelihood of being pathogenic and very low fitness scores. And we have, in the other hand, another group that has a conflicting towards benignity that shows higher fitness scores, but low likelihood of pathogenicity. So this is very exciting to see this trend when we look at all the cohorts. So as an example, this is an ongoing genotype-phenotype analysis we are doing with the Mayo Clinic, and the same pattern shows here. And they are actually going to look at the correlation between the phenotypic data they have of these patients to the fitness scores that we provided them. So the same trend happened here. A group of variants has low fitness scores, high probability of being pathogenic, and some have very high fitness scores and low prediction of being pathogenic. So I'm really excited to hear back from this group and see if there's any correlation between severity, onset of disease, maybe oxalate level, maybe severity of the kidney damage. So this is something that is going to be available in the manuscript very soon. And I want to show you actually a cool case about the infantile severe form of PH1. So this group actually did the whole genotyping of this family that was affected by the infantile severe form of PH1, where the boys on the individual 16 and 17, they died 4 and 5 months of age, 2 different pregnancies. And both of them carry 2 mutations that we have scores for in the residues 173 and the residues 223. And it would be really interesting to know if our map is actually sensitive enough to segregate or tell apart which mutations could be the contributing to the severe form. So it would be really nice to see any phenotypic data of this family to make sure that if our map is sensitive in this sense, then we will have some type of evidence that the mutation on the residue 173 could be the one actually leading to the worst prognosis. So when they sequence and genotype this family, one side of the family, the maternal side of the family, had the 173 mutation, and the paternal side of the family had the 223 mutation. And this study actually provided us with phenotypic data saying that only the heterozygous people for the 173 arginine variant actually experience kidney stones in their lifetime, while the other heterozygotes are completely symptomatic. So this is indicative that our map could be sensitive to detect severe form of disease. So finally, I was able to show today that our test can separate variants based on function, that our method detects important residues either for PLP binding, dimerization, and we detect PH1 pathogenic variants so far having low fitness scores. We provide also new evidence, experimental evidence for VUS variant in PH1 cohorts, and these results together show that our method is not only accurate on small scale, but yes, when multiplex in different background contexts and different cofactor contexts. So as next step, I wanted to optimize the separation of the minor allele. Maybe I'm going to culture this for a long time, make sure that the yeast get enough doublings with the minor allele defect, and analyze this map in the presence of the cofactor PLP. And I want to finalize the correlation analysis between disease type, severity, and fitness scores. I would like to thank my lab, especially Zidai and Anna that helped me with experiments, Johan that helped me figure out the data analysis that I did all by myself, Atina and Marinella that helped me with sequencing, and of course, Fritz, that is the most amazing supervisor that I ever met, and I cannot thank him enough. Thank you.
A wonderful presentation, Adrini. So while we wait for question from the audience, so do you think that there could be differences of variant effect with the yeast complementation assays versus the human cell line assay?
Possibly. I haven't tested using a human-based assay, but because we know that in humans, there are also some level of interaction with other proteins from HXT. So maybe in a human-based assay, we could explore that site. But because our method was very designed towards the biochemical enzymatic activity, so far it has shown that it can really separate functional and non-functional.
So I can ask one more. So in your slide where you showed the map, it looked like some of the nonsense mutants weren't deleterious, that weren't deleterious were at the very end of the gene. Could this be some effect of gene somehow escaping NMD?
Yes, escaping NMD. So actually what we hypothesize is that once there's a lot of frame shifts in this region, so maybe having a frame shift that is going to elongate the protein so far off might have a worse effect than some other deleterious mutation that would just stop the function at that region. So it's a hypothesis that we have, but also we know that this site, this red box, is where substrate binds. So maybe yeast is actually evolutively acquiring mutations that help bind the substrate better. So they're very nice questions about it. And this is a really interesting questions, and I really want to see the answer to the deepest level, the biochemic structure of the minor protein.
So I don't see any other questions, but I see we are at the top of the hour. So I would like to thank both Megan and Adrini for excellent presentations. Thank everyone for attending. And remind that next month we have again two fantastic presentations from Willow Coyote Maetes at UFC and from Rayon Zhong from Harvard Medical School. So, and just as a reminder, the talk is recorded on YouTube, so feel free to go back and check any part of these. So thanks everybody, and see you next month.
Thank
Bye.
you.
Thanks.